Okay, the last brave souls. I'm Mike Telkowski. I'm a faculty member here at the CHGR and Harvard Medical School, and I'm going to introduce Ian Blumenthal, who came to my lab after a bachelor's in bioengineering. Ian has been following up on many of these gene discoveries with functional analyses, and he's going to talk to you about transcriptomic studies, the 16p11, and efforts that we're making to go kind of beyond the gene discovery effort to really try to understand what these loci are doing at the level of the transcriptome. So here's Ian.
Good afternoon, everyone, and thank you very much to the consortium for having me here today. There we go. So we've heard a lot about 16p today, and I'm going to sort of gloss over the basics. We've heard that copy number variations are very important or have been identified as a major contributor in autism, and that 16p11.2 specifically accounts for approximately 1% of all ASD cases. The mechanism of deletion or duplication of this particular region, namely non-allelic homologous recombination of the region's flanking segmental duplications, leads to very consistent breakpoints in affected genes, as we've seen in a number of the other talks today. But a number of questions remain due to the variable phenotypic outcomes that we see in the disorder. Deletion generally has a worse phenotypic presentation than duplication, but neither abnormal copy state is fully penetrant, and both have been highly associated with a wide spectrum of developmental, psychiatric, and anthropometric traits aside from ASD, a number of which I've highlighted here. The fact that reciprocal dosage change at the DNA level can lead to similar disorder opens the door to a number of very interesting questions as to the phenotypic effects of reciprocal expression changes at the RNA level. Two zebrafish studies published last year, including one from the CIV group at the Whitehead, some of their data we just heard about, as well as a study from the Katsanis group at Duke, identified anatomical phenotypes due to the under and overexpression of individual genes within the 16p region, suggesting both that single genes in the region can contribute elements of phenotype, and that deletion and duplication can have different, if related, effects. However, existing studies have yet to reveal the underlying mechanisms driving disease in the disorder and leave a large portion of the variable phenotypic expressivity unexplained. So as we approached the study, we had a number of primary questions that we were hoping to answer. The first is, what is the transcriptional impact of genomic disruption at the 16p locus? The second is, are there gene expression patterns associated with the phenotypic heterogeneity that we see in the disorder? The third, are there overlapping or independent networks dysregulated by a direction of dosage imbalance? And finally, more on a methodological level, are lymphoblastoid cell lines an appropriate model for neurodevelopmental traits? So to investigate and answer some of these questions, we performed two parallel expression experiments. The first one was in 35 lymphoblastoid cell lines from 7 families, seen here. They were sourced from Agree, and we had 4 deletion families and 3 duplication families. And these families were selected not just for presence of 16p copy number variation, but because each of them harbors multiple affected individuals as well as some level of discordance. You can see here on the top left that we have a family with 2 affected individuals, but only one of them has CNV. And on the bottom left is a family with 4 individuals with copy number variation, but only 2 of them are affected. The question remains, however, as to how relevant a model are lymphoblastoid cell lines to the study of neurological disorders. And as you can see here, an example from PRRT2, a gene found within the 16p region, which has almost 3 orders of magnitude greater expression in human brain than it has in lymphoblastoid cell lines. This pattern of expression is not unique to this gene, even within the 16p region itself, and given the strong influence of 16p11.2 on neurodevelopmental and neuropsychiatric disorders, it is likely to extend to genes influenced by 16p dysregulation as well. Finally, I'd like to make the point that not all expression is functionally equal. Here we have an example of this from TCF4, a gene implicated in Pitt-Hopkins syndrome, schizophrenia, and more recently in ASD as well, that while at a gross level, the expression of TCF4 in human iPS-derived neural progenitor cells from a different study, that the total level of expression in these NPCs is about half that of the total level of expression in lymphoblastoid cell lines. These levels of expression are driven by tissue-specific transcription start sites and result in different isoforms in different proteins. We can see a similar example in another recently ASD-implicated gene, CHD8, down here at the bottom, which further highlights the differences between these two tissues. So to address some of these issues, and as I mentioned a moment ago, we performed a parallel expression experiment in the cortex of 16 mice in a mouse model with copy number variation of the 16p syntenic region. And we heard a little bit about these mice a little earlier today. They were developed by Leah Mills's lab down at the Cold Spring Harbor Laboratories, and they allowed us to create a fully gender and genotype balanced study design in the tissue most relevant to the disorder. So as a very quick overview of our methods, we generated strand-specific polyase-selected RNA-seq on 16 mouse cortex samples to a depth of 40 million reads per sample, and 35 human lymphoblastoid cell line samples to a depth of 60 million reads per sample across 2 biological replicates. We performed our alignments with Top Hat 2 against the HG19 and RefSeq references, and we included ERCC RNA standards for downstream quality control and expression thresholding. So starting out, we found relatively quickly that existing statistical packages for differential expression in RNA-seq inadequately modeled the complexities of our family-based study design, and we elected to use general linear mixed models and followed up with permutation to generate empirical p-values, which you can see on the top right fit the expected distribution quite nicely. And also, given the strong hypothesis for reciprocal effects, we opted to use 2 complementary modeling approaches, the first of which was contrast comparing wild type to deletion or duplication, and was very good at identifying strong effects. And the second was a model which identifies linear effects across the 3 genotypes, allowing for the incorporation of all of our data and improving our power to identify smaller but consistent reciprocal changes. So before we began modeling, I mentioned that we were very interested to gain insight into the relevance of lymphoblastoid cell lines in the study of neurodevelopmental traits, as well as to gauge how much overlap we could expect to see between our different cohorts that we designed. On the right, we can see Spearman rank correlations of the averaged expression values between each genotype, and as anticipated, the correlation in expression between samples of the same tissue was quite high. However, the correlation in expression between the mouse cortex and the human LCLs was not very good. Interestingly, when you throw in a set of human iPS-derived neural progenitor cells, again from a different study, you find that the correlation in expression between the mouse cortex and the human NPCs is about on par with the correlation in expression between the human LCLs and the human NPCs. With regard to whether or not the genes expressed in each of these tissues are the correct genes to be studying, we assembled a composite list of known ASD loci from Safari and Autism KB, and we found that many more ASD genes were expressed in the mouse cortex than in the human LCLs. However, there was a substantial number of genes previously associated with ASD expressed in the lymphoblastoid cell lines. So the first question we had about the effects of abnormal copy state is what expression changes are occurring within and around the CNV itself. Even without any statistical modeling, we can see relatively even expression changes throughout the entirety of the region, with an increase to about 1.5-fold expression in duplication and a decrease to about 0.5-fold expression in deletion, with intermediate expression changes in the flanking segmental duplications, which I think it's important to note were not present in the mouse model, and a number of positional effects in the outerlying areas in both human and mouse, including a few that overlapped between the two. So moving genome-wide, you can see in these Manhattan plots of p-values across both the mouse and human genomes that there is nothing in the genome which produces a reciprocal effect anywhere near as strong as the effect that the 16p region does, and that most of the genes which remain significant after multiple test correction emerge from the 16p region itself. So taking the nominal genome-wide signals, we wanted to ask the questions as to which genes' expression changes correlate the most closely with the genes found within the 16p region. To do this, we used weighted gene correlation network analysis to highlight clusters of genes with highly correlated or anti-correlated expression between the different conditions. And we found that, as you can see here, that in both human and mouse, a majority of the genes clustered closest with the 16p genes were anti-correlated with the expression changes of the genes in the region. While there was little overlap between the two gene sets which clustered with the 16p genes, the consistency of the correlation pattern suggests that one of the downstream effects of the dysregulation of the 16p genes is an overall downregulation of other genes throughout the rest of the genome. And in fact, what we find is that this trend was also reflected in the genes outside of the modules, as seen here in the mouse, with deletion resulting, with the majority of the genes dysregulated in the mouse being in deletion being overexpressed, and the majority of the genes dysregulated in duplication being underexpressed. Looking at some of the gene ontologies and pathway annotations for our nominal results, identified a number of dysregulated pathways which overlapped between both the human and mouse data sets. I've listed a few here which emerged as some of the top hits, including histone methylation and actin filament organization, the latter of which has been shown to play an important role in synaptic transmission and long-term potentiation. And down here on the bottom, I've selected a number of the top hits that were specific to the mouse cortex, which included a number of terms which had been previously implicated in autism, including cell adhesion and neuron differentiation. I'd also like to add that while the results missed significance for multiple test correction, we also saw intriguing evidence for the dysregulation of genes involved in adipogenesis and sugar metabolism in the mouse cortex, hinting at some of the mirrored BMI phenotypes that have also been seen in the disorder. So taking our about 900 nominal genes in the mouse with a reciprocal effect and running them through DAPL yielded a direct interaction network of about 280 of those genes, 6 of which were 16p genes and 23 of which were loci previously implicated in autism, seen in red and blue here respectively. And this is a subnetwork of that larger network that I just told you about, which was particularly interesting for its high density of ASD-related genes seen in blue and 16p genes seen in red, as well as a high degree of interconnectivity between them and novel loci. We performed a similar analysis in the human set of about 550 reciprocally affected genes, and we found a network of about 100 of those clustered together quite nicely. I have a pop-out box here of a subnetwork of those that we thought were particularly intriguing. It's been highly implicated in nucleosome assembly, and it interacts indirectly with the 16p gene MAPK3 through the previously autism-associated gene HDAC4. These histone genes were interesting not only because a few of them have been previously implicated in autism, but because they and a few other genes in the network all originate from the same locus on chromosome 6. And in fact, that when we computed enrichment for the dysregulation of that chromosome band, specifically 6p22, we found that the enrichment was much greater than one would expect by chance. With this in mind, we looked at a database which curates data describing the three-dimensional conformation of chromatin in the cell. We found that there was a high correlation between the dysregulated region 6p22 and 16p11.2. You can see here in this graph, which shows the distribution of p-values across chromosome 6 on the top of a heat map showing the degree of correlation with 16p11.2, with red representing a greater likelihood of interaction. This is not definitive evidence for a physical interaction between the two, but it is certainly sufficient evidence to warrant additional study and to pose additional questions as to the impact of C and V on nuclear organization. Another question we hoped to answer was whether the dysregulation of 16p11.2 propagated to other known ASD loci, and I've hinted at it throughout my talk that it has, but here are the actual numbers and enrichment values. And we found statistical enrichment for the dysregulation of known ASD genes in deletion in both human and mouse, and across both genotypes. And we found in total 155 unique ASD genes dysregulated in the mouse cortex at a nominal level, and 76 were dysregulated in the human LCLs, with about a quarter of the human total overlapping between the two models. Among these hits were a number of genes that we were particularly interested in due to their recent identification in a series of exome and translocation sequencing studies from last year. And of particular interest, we saw convergence of dysregulation on ASD genes which address multiple hypotheses for the pathogenesis of ASD. Namely, we see dysregulation of genes which function at the synapse, represented in this chart here by Nerexin 1 and Nerexin 3, as well as dysregulation of some of these more recently identified chromatin modifiers and transcription factors, CHD8, EHMT1, SATB2, and finally a number of genes involved in more classical forms of intellectual disability such as FMR1. So making a direct comparison with previous transcriptomic work in ASD, we found substantial overlap between our study and a previous study of expression changes in the brains of idiopathic, non-syndromic forms of autism from the Geschwin group at UCLA, finding dysregulation in our study of about a quarter of the genes that they found in theirs. Outside of specific genes, we also found convergence on very similar pathways, including dysregulation of pathways involved with synaptic function and axon guidance, again suggesting convergence on underlying pathways from multiple forms of ASD. And to conclude, we found that within the 16p11.2 locus that there was a very clear gene expression signature, and that the expression signature which emerged from the locus itself was by far the strongest signal that we could see in the genome, suggesting a distribution of relatively subtle dysregulation and highlighting the need for even larger sample sizes and greater power than we were able to achieve in our study. And finally, that the genes dysregulated by 16p11.2 C and V converge on common pathways containing known ASD loci. I'd like to thank everyone who helped me work on the study, especially my mentors, Mike Tolkowski and Jim Gusella, and I would like to thank the Simons Foundation and the Nancy Lurie Marks Foundation for their help in supporting this research. I'm open to questions. Thank you.
You know, one interesting thing I learned from the Sweden, the Karolinska Institute, you know, just a few years ago. I still scratching my head because, you know, they use the transcriptome technique and they isolate the gene in asthma. You know,
in what?
In asthma.
Mm-hmm.
In therapy-resistant asthma. It turned out to be a surprising, seemingly irrelevant gene called the beta-tase receptors, TAS. The reason I mention that is, you know, I look back into the previous lecture. If you look into the KIF22, which is a kinesin family,
Mm-hmm.
and it's just not, you know, related to the ASD, but also related to muscular skeletal dysplasias. So I wonder, you know, when we pick the candidate gene or the gene you're in interest, what is the criteria, or do we miss something that may be as significant clinically as the Karolinska studies when they surprisingly, unexpectedly, you know, pick up the beta-tase gene and they find tasting something bitter can relax the, you know, vasoconstriction of the sumuscle in the trachea.
So
we might miss something in the ASD.
Mm-hmm. I hear you, and I think coming into the study, we tried to be as agnostic as possible with respect to which genes within the 16p locus are really driving the pathogenesis of ASD. And we tried not to really sort of cherry pick which ones we were the most interested in, and sort of looked at what is dysregulation of this region doing genome-wide, and trying to gain insights into ASD from those networks that we were able to generate. Does that answer your question?
Yes. My fundamental, you know, skeptical, you know, thinking is that when a candidate gene, they have multiple functions involved in multiple systems,
Mm-hmm.
how can we qualify, you know, that gene is significant, you know, in the ASD? That's it.
I think that's a very fair point, and I think with something like 16p, we very directly see that the same copy number variation can lead to a wide variety of phenotypic presentations.
Yeah.
They can finish these up over the bar.
Thank you.
Statistical arguments are very different than biological hypotheses.
Yeah, but actually, if you talk to our common friend, you know, Jim Gusella, you know, he told me the interesting story, the Huntington gene, the Huntington, you know, they have moonlighting protein functions that I was surprised. That's what I think. You know, I try to link up Jim's, you know, idea and the Karolinska, the beta-tase receptors, and the KIF22.
Great part of the discussion.
Thank you for your question.
Thanks for everything.
Thanks, guys.